Hello, and welcome to our FDA Animal Biotechnology webinar on the Bioinformatics review of next-generation sequencing data. My name is Alexis Norris, and I am a bioinformatics reviewer in the Division of Animal Bioengineering and Cellular Therapies in the Office of New Animal Drug Evaluation at the FDA Center for Veterinary Medicine. I am joined today by my fellow bioinformatics reviewer, Ashley Cook. Together, we will present an overview of the goals of our bioinformatics review of next-generation sequencing data, abbreviated NGS, in support of the molecular characterization of intentional genomic alterations, abbreviated IGAs, in animals, and the process for electronically submitting NGS data using FDA's precisionFDA platform. There's an increasing number of developers using next-generation sequencing as a tool for product characterization for IGAs in animals, notably for the confirmation of the IGA and identification of any unintended alterations. Next-generation sequencing allows for screening the genome in a single assay, providing valuable information for FDA's review of the IGA. As part of the review process, CVM performs an independent analysis of the NGS data submitted by the developers. This webinar provides an overview of that analysis and describes how to submit large NGS datasets to CVM that support the molecular characterization of IGAs in animals. Our agenda today will cover information about the Division of Animal Bioengineering and Cellular Therapies, abbreviated DABCT, an overview of the review, an overview of the common types of NGS data, an overview of what our analysis looks like, and finally, the process for sharing data using precisionFDA. The Division of Animal Bioengineering and Cellular Therapies is within FDA Center for Veterinary Medicine's Office of New Animal Drug Evaluation, abbreviated ONADE. The division reviews animal cells, tissues, and cellular tissue-based products, abbreviated ACTPs, and intentional genomic alterations in animals. We collaborate closely with the ONADE, with other CVM offices, with other FDA centers, and other FDA programs. Our division has expertise across a wide array of scientific disciplines, including molecular and cellular biology, bioinformatics, genetics and genomics, chemistry, stem cell biology, animal science, and veterinary medicine. The Animal Biotechnology team evaluates IGAs in animals. IGAs are generated using modern molecular techniques, for example, recombinant DNA techniques and CRISPR-Cas9 genome editing. IGAs may include random DNA sequence changes or targeted DNA sequence changes. An IGA can impact gene or protein expression to produce a desired phenotype. And these IGAs have a variety of applications, including industrial, agricultural, medical, and ornamental. FDA uses a risk and science-based review process focusing on safety to the animals containing the IGAs, safety to the people or animals that consume food from the animals, and risk to the environment, in addition to confirming that the IGA does what it claims to do. The approval application review process for risk characterization consists of 6 major steps and starts with product characterization. NGS data is often used by developers and their product characterization to confirm the IGA is present in the animal and screen for unintended alterations. For more information about the other steps, visit our CVM Animal Biotechnology Products Resource Center website to view previous webinars. CVM performs an independent analysis of the NGS data submitted by the sponsor. As mentioned on the previous slide, the NGS data answers 2 major questions. First, do the animals have the IGA? And second, are there any unintended alterations that might impact safety? Specific products have specific questions, which is why we ask developers for information about the methods they use to generate their product. For example, if a plasmid was used, did the plasmid integrate into the animal's genome? Or if an embryo editing was used, do the animals have low-frequency mosaic alterations at the target or off-target sites? Common NGS technologies are grouped by the length of the sequencing reads they generate. either short reads such as Illumina or long reads such as Oxford Nanopore Technology and PacBio. Additionally, the NGS assay might assess the entire genome, referred to as whole genome sequencing, or only specific regions. These regions can be targeted using hybrid capture baits or PCR amplification approaches. The first step of our analysis is to identify the variants in all samples for which data was submitted, both in animals with the IGA and comparators. We look for both short variants and larger structural variants. Short variants include single nucleotide polymorphisms, or SNPs, and short insertions and deletions, abbreviated indels. Structural variants include larger insertions and deletions, as well as genomic rearrangements like inversions and duplications. To identify the variants, we align NGS sequencing reads to a reference genome, which could be either the publicly available version for the species or a sponsor-provided genome assembly. If exogenous sequences were used to introduce the IGA, those sequences should also be included in the reference genome for alignment. The second step of our analysis is to remove variants that were likely inherited. The presumed inherited variants could be based on developer-provided comparators, for example, unedited parental cell line, unedited dam and sire, unedited sibling, or other wild-type animals. We also use public databases such as dbSNP and the European Variant Archive, or EVA. The goal of the third step of our analysis is to remove artifactual variants due to technical error during the sequencing or alignment. One step is to visually compare the read alignments to the reference sequence. We look for a clean sequence in the comparators compared to animals with the IGA. We also filter for variants with an allele fraction consistent with the method used to introduce the alteration. In the case of edited cell lines, the variant should be heterozygous or homozygous. We also remove sequencing artifacts due to sequence context, including simple repeats like homopolymers, and we ensure the variant is found without strand bias, meaning that it's present on both the forward and reverse sequencing reads. Additionally, we account for mapping errors, which are identified by low mapping quality and low sequencing coverage. And finally, we exercise caution in repetitive regions, and we consider results from orthogonal methods provided by the sponsor, if available. A common orthogonal method is Sanger sequencing of specific regions of interest. The fourth step of our analysis is to determine if the confirmed variants occur in or near predicted cut sites. This can be performed using an in silico homology-based algorithm that considers off-target sites resulting from mismatches and bulges between the guide RNA and the host genome. We also consider additional empirically nominated sites, if available, from sponsor-submitted biochemical assays such as SiteSeeker CircleSeq and cell-based assays such as GuideSeeker ChangeSeq. The final step of our analysis is to use available genome annotation data to inform hazard characterization of confirmed variants. We consider databases of genes such as Ensemble and NCBI, repetitive elements, including mobile elements using repeat masker, conservation across species such as phyla, phos, cons, and GERP, and regulatory elements when available, such as chromatin and CPGs. I'll now hand it over to my colleague Ashley Cook to walk us through the process for electronically submitting NGS data using FDA's precisionFDA platform.
Thanks, Alexis. Now I will share with you information about precisionFDA. precisionFDA is a cloud computing platform developed by the FDA that is appropriate for regulatory data. It is a secure platform with a FedRAMP moderate rating. precisionFDA can be used to share and analyze data. However, it is not intended for permanent storage. There is no cost for use of this platform. Now we'll go through an overview of the steps to share data with CVM using precisionFDA. Step 1 is to create a precisionFDA account. This is done by simply completing the request form and setting up multi-factor authentication. The next step is to notify CVM that you intend to share data. This is done by sending an email to cvm_precisionfda@fda.hhs.gov. We will then create a secure space for you to upload the data, and you will receive an email to activate the space, which involves simply clicking on the URL in that email. After you've activated the space, you are able to upload files to the space's shared area. Finally, in your e-submit or submission, include information about the files shared through precisionFDA, including the precisionFDA space URL, the file names, their unique precisionFDA file IDs, and information about the file, for example, the sample identifier. We'll now do a demonstration walking you through these steps on the precisionFDA website. When you go to the precisionFDA website, in the top right corner, you will see a button called Request Access. Clicking that takes you to the form to request a precisionFDA account. I've boxed the fields that are important to complete: your name, email, and use. You then click Request Access to precisionFDA. If you do not receive an email provisioning your account within 2 business days, please contact CVM_PrecisionFDA at fda.hhs.gov and we will look into it. After your account is set up, email our CVM precisionFDA inbox to create a space for you. You will then receive an email with a link to activate the space. Once the space is activated, you are able to upload files to share with CVM. I will now walk you through how to upload the files. To navigate to your space, you first log into your precisionFDA account using the Login button on the top right corner of the precisionFDA webpage. Once logged in, you can click on the Spaces icon at the top of the precisionFDA site to view your spaces. For this demonstration, I've created a space called Demo Review 2. Clicking on the space's name then takes me into the space. The space includes both a private area and shared area, which you can switch between using the tabs. The private area is accessible only to you and users you add. The shared area is accessible to CVM. Thus, to share files with CVM, you must upload them into the shared area. Both areas have the same sections, which are on the left side of the page and include files. As mentioned earlier, precisionFDA is also a computing platform. The apps, workflows, and execution sections relate to computing. To upload files, you simply click on the Files section on the left side of the page and then click the Add Files button. In the case of larger files, such as 5 gigabytes or more, and in the case of tens of files to upload, we recommend using the Command Line Interface tool, or CLI. This tool is available for download from the precisionFDA website, and there are versions for Linux, Mac, and Windows operating systems. For security reasons, uploading files requires an authorization key, which expires 24 hours after its generation. You can generate the CLI key in the Assets section of precisionFDA. Here, clicking Assets will take you to that page. You can also access the Assets section by clicking on My Home icon at the top of the precisionFDA web page and then clicking on Assets on the left side of the screen. To generate the CLI key, click on the Generate CLI Key button. This produces a pop-up with text that you copy and paste to use as the key in your upload command with the CLI. The precisionFDA documentation includes commands for many common scenarios, including uploading a single file to the space, uploading multiple files, and uploading an entire folder or directory of files. If you encounter any issues, we are here to help you. Simply email our CVM precisionFDA inbox. If the data is accessible with a URL, an option is to directly transfer the data to precisionFDA using the URL. For this, we have an app called URL Fetcher. On the precisionFDA, tools are called apps. You can find apps under the Apps section. For the URL Fetcher app, you provide the URL when you run the app. If you would like to use this method, we are happy to help you. Simply email our CVM precisionFDA inbox. There is no one-size-fits-all with NGS data. Questions and data expectations vary by product, and NGS is one approach to addressing molecular characterization. We are ready, willing, and able to analyze data that you submit. We are happy to meet with developers to discuss the questions and needs for their product and to share information about how to submit data. We encourage developers to have conversations with us early in the process, and we can walk you through using precisionFDA. One of the initiatives outlined in the Plant and Animal Biotechnology Innovation Action Plan is the Veterinary Innovation Program, or the VIP. The goal of the VIP is to facilitate advancement in the development of innovative animal products by providing greater certainty in the regulatory process, encourage development and research, and supporting an efficient and predictable pathway to approval for ACTPs and IGAs in animals. It is available for certain IGAs in animals and ACTPs that provide a benefit to human or animal health, animal well-being, or enhanced food production. The VIP offers many important benefits to help sponsors and developers through the regulatory process. These benefits include access to a toolkit with helpful resources to aid you in the review process, frequent interactions between CVM and sponsors, a dedicated review team, opportunities to discuss alternative data strategies to meet requirements, senior CVM management involvement, feedback on assay development, feedback on submissions prior to submission and after conclusion of the review, the ability to stop the clock during the review process to submit data to address deficiencies, and hands-on how to help for post-market responsibilities. We welcome any feedback and questions you may have on this webinar. Please submit these to FDA-CVM-Animal-Biotechnology-feedback@fda.hhs.gov. This slide contains additional contact information you may find helpful, including the email address for general inquiries, askcvm@fda.hhs.gov, the email address for biotechnology-specific inquiries, askcvm-biotech@fda.hhs.gov, the email address for precisionFDA-specific inquiries, cvm_precisionFDA@fda.hhs.gov, and the Animal Biotechnology Team Leader, Adam Moyer's email address if you wish to contact him directly. Thank you for your attention. We hope you found the information in this webinar to be helpful.